Hello, everyone, and welcome. Glad you could join us today for a Nucleai Academy session on AI spatial biomarkers and immuno-oncology. My name is Byron Lawson. I'm the Vice President of Business Development and Strategic Partnerships here at Nucleai, and I'll be your moderator for the webinar. Before we get started, just a few quick housekeeping notes to ensure a smooth experience. Today's session will run approximately 60 minutes. We'll begin with a 40 to 45-minute presentation, followed by live Q&A. The session is being recorded, and we'll share the recording with all registrants after the webinar. To minimize background noise, all participants are currently on mute. If you have any questions during the presentation, please submit them via the Q&A box at the bottom of your screen. We'll do our best to address as many as we can during the Q&A portion, but not during the presentation. We'll also be launching a few poll questions throughout this session. When they appear, we encourage you to take a moment and respond. We'd love to hear your input, and we'll share the answers with the audience. If you encounter any technical issues, try refreshing your browser or rejoining the session. You can also send a message in live chat, and our team will be happy to assist. Now, without further ado, I'm pleased to introduce our speaker, Dr. Atay Markovits. Dr. Markovits is a medical professional and researcher. He earned his medical degrees with honors from the Hebrew University and completed his PhD at Tel Aviv University, where he focused on immunotherapy response and resistance mechanisms in melanoma. At Nucleai, Dr. Markovits serves as Director of Biomedical Research, leading efforts in cancer biomarker identification using spatial biology. His recent work involves developing advanced analysis pipelines for multiplex imaging and leveraging deep tumor microenvironment profiling to drive biomarker discovery and enable precision medicine. Atay, the floor is yours.
Hi, thank you, Byron, for this kind introduction. In today's talk, we're going to discuss how we can use the tumor metabolic profiles to understand the relation in immune response and resistance, and how can we use that to develop better predictive biomarkers for immunotherapy response. I will start with presenting our motto at Nucleai, that we're an AI special biomarker and diagnostic company. We use our AI-powered tissue structuring algorithms to extract insights from all kinds of imaging modalities, whether it's H&E, IHC, and multiplex imaging, with the goal of to develop better diagnostics to improve patient care in this era where there are multiple choices for treatment for each patient. We believe that the use of AI-driven spatial biomarkers in treatment decisions is inevitable. And the reason that we believe it is, first, because it's already happening. There is a growing adoption of spatial biomarkers in drug development, whether it's the seminal work from AstraZeneca that showed the importance of AI-based protein scoring, and also the calculation of spatial proximity scoring in order to improve patient selection for HER2 ADCs. Also, we have our own trial that we use our technology to select patients for a phase 1 clinical trial, where we use AI to help pathologists score multiplex IHC. And there is also a clinical trial that uses multiplex imaging, low-plex assay by a Koya PhenoImager to select patients as well. Another reason is that there is a growing need for biomarkers that will help clinical decision making. There are currently more than 600 novel therapies in drug development pipeline, a lot of antibody drug conjugates, immunotherapies, cancer vaccines that are really revolutionizing the treatment in cancer patients and helping them to receive better treatment outcomes. But then in the era of the multi-treatments, there are several questions that clinicians need help answering. First is, how can we give the patient the best chance to respond or not to relapse while minimizing toxicity? So from the approved drugs, which are usually not compared head to head, how can we best navigate the patient to the best treatment that he can get? And also in this era of a lot of clinical trials that are ongoing, how can we direct patients to the clinical trial that they will most probably benefit from? And most of these cancer and next generation oncology treatment, they need deep spatial insights in the protein level to really accurately predict the response, whether it's within the immunotherapy field, that we know that it doesn't just matter which cells do we have within the tumor, but their spatial organization is critical for understanding the treatment response. Or whether it's the antibody drug conjugate, that we know that there is an effect for the bystander effect, and to really quantify it, you need the spatial biology to identify the tumor cells which are negative for the target protein, but are in proximity to positive tumor cells, that their killing can affect their neighbors and thus help responses. And also within the multispecifics, which targets proteins that are co-expressed, and you need to really understand the spatial relationship between the two targets in order to maximize patient responses. So all of this treatment can benefit from spatial biology. And we know that pathologists sometimes, it's a difficult task for them to identify exactly the spatial patterns that are within the tissue. And we believe that AI-based biomarkers can really help and move this field forward. The third thing that enables the adoption of spatial biomarkers based on AI is that there have been three really technology shifts that enable the extraction of the spatial insight that we need to make an accurate prediction or to really understand disease biology. And these shifts were the digitization of pathology, advanced imaging techniques such as multiplex imaging or spatial transcriptomics, which really allows us to get deeper insights into the tumor biology and to develop better diagnostics. And also AI advancements such as deep learning and foundation models that allows us to accurately procure these insights from these advanced imaging techniques. And with this, I want to stop now and to go to our first polling question, which is, in which area of clinical decision making do you see biomarkers as the most critical? And this is a multiple choice, so you can answer a few questions, and the options are identifying the right patients for therapy, patient selection or certification, predicting likelihood of treatment response, monitoring treatment efficacy and disease progression, reducing the risk of adverse event toxicity, or guiding regulatory approval and clinical trial success, or other. So please vote. And as we can see from these answers, there are multiple choices, and most of you agreed that in almost all of the above, biomarkers are important, but especially within the identifying the right patient for therapy, which is patient selection and certification. And with that, we want to go to our next polling question, which is, which platforms are you currently using for biomarker discovery? And again, this is a multiple choice, and the options are genomics, transcriptomics, immunohistochemistry, H&E, multiplex immunofluorescence, spatial transcriptomics, or other. And we can see the result that most of you use immunohistochemistry, but there are also representation for all other modalities, including MIF, H&E, and transcriptomics. Now we go to our last polling question for this question, which will be, are you currently applying AI tools for biomarker discovery or for clinical diagnostics? And this is a single choice question, and the options are yes, primarily for biomarker discovery, primarily for clinical diagnostics, for both areas, or not yet but planning to, or no. We'll have the results shortly. And we can see that it's interesting, but no one answered no, and most of our participants use them either for biomarker discovery, clinical diagnostic, both, or planning to use them in the future, which is great. So with that, we'll go to the next slide. And in this talk, we're going to focus mostly on multiplex imaging. We also have our own tools for H&E and immunohistochemistry. But in this talk, we'll talk about multiplex imaging because it's a very powerful tool for biomarker discovery. And the reason for that, it can really allows us to capture tissue architecture and the cell interaction and functional states within this architecture. And moreover, proteins are what are used to classify cells. They are the gold standard for classifying cells, so we can really accurately define both the cell subtype and phenotype, and to capture that within the tissue and the relations regarding the different tumor areas. And as our understanding of spatial biology and disease complexity grows, we need more and advanced imaging tools that can capture these complex biomarkers. And we can see from this meta-analysis that compared between different biomarkers for immunotherapy response. And you can see here that the best area under the curve was immunofluorescence-based methods for anti-PD-1 response. And we can see also from this work that was published in Nature in 2020 that showed the importance of TLSs and their association with immunotherapy response. And to actually identify the mature TLSs, we actually need a combination of markers that allows us to identify B cells, follicular dendritic cells, and T cells. And in a follow-up work, what we show that it's not enough that you can identify the TLSs. The cells that are within the TLS and their functional states are also critical for better prediction. And in this case, it was shown that if you had a high CD4, CD4 FOXP3, if you had a lot of regulatory T cells within the TLS, it was associated with a worse outcome versus patients that had low regulatory T cells. So really, if we want to identify regulatory T cells within the TLS, we already need a combination of 4 to 5 markers to do that accurately. And this is where multiplex imaging can really help us with the development of biomarkers. So in Nucleai, we put a lot of effort to develop a pipeline that can be applied robustly and generalizably to really capture and get these biological insights from multiplex imaging. And our pipeline is a combination of both machine learning and deep learning based algorithms. Usually start with QC of images and markers, where we developed also machine learning based techniques to do that automatically. Then for structuring the multiplex imaging image, then we have a combination of 4 deep learning algorithms that we developed in-house, which can help us identify defects and exclude them, perform cell segmentation and identify the cells, to perform intensity normalization, which we developed a novel deep learning-based algorithm for MIF normalization, and then a deep learning algorithm for single-channel protein quantification, which I will go deeply into in a minute. And once we have for each cell, for each identified cells, the quantification of each protein that we have in the panel, we can do all sorts of downstream tasks, such as the cell typing and cell state phenotyping, identification of spatial neighborhoods, and area modeling. And then we can combine all of these inputs for feature calculation and spatial analysis. So in this talk, we're going to focus mostly on the single channel protein quantification, because this, coupled with the normalization, is really what powers our analysis to accurately define the cell types and their phenotypic state, which is critical to have meaningful biological features, spatial features that can be used for biomarker identification. So there are several current limitations with MIF analysis methods. Most of them are based on mean marker intensity within segmented cells. And that could be really problematic. It can be affected by segmentation errors. It can be affected by variability in intensity within the slides and between slides, by background noise that can affect our signal, image artifacts, and cellular projection. And because all of these can really affect the quality of the signal that we get, then downstream analysis, which is based on this signal, can be really problematic. Both manual gating or clustering-based methods can be laborious, user-dependent, inaccurate, and not really transferable between panels and cohorts. This is why in Nucleai, we harness the capabilities of deep learning to overcome this limitation. And convolutional neural networks really revolutionize computer vision, and it's a great for the task of protein expression quantification, as it really learns the protein expression patterns within the image to better classify cell types. In this case, for example, we can see it managed to distinguish between not so easy to distinguish signal. We can see that there is a lot of overlap, but because it learns the nucleus and the contour of the membrane and the protein expression pattern, it enables us to accurately profile even within these dense tissues. So really, we use the power of CNNs to develop our own signal channel binary classifier, which receives. We identify the cell, then we crop a multi-channel tile around each cell. We can then decompose this multi-channel tile to single-channel tiles, which are inputted into our single-channel binary classifier. And then for each combination of cell and marker, we get a positive predictive probability. And these probability can be automatically threshold for phenotypic marker quantification, or can be used for cell typing. We use supervised cell typing, where we predefine the expected marker expression within each cell type based on the panel composition. We also validate it in unsupervised manner to make sure that we do not miss a certain phenotype, but we found it to be a lot more accurate than methods that use unsupervised approaches. And what this deep learning binary classifier was trained on hundreds of thousands of single channel annotations, where it was marker agnostic. So it got a single image, a single channel tile centered around the cell. And then it got only either a positive or negative label. And we use a very wide variety of markers, both of nuclear, membranous, cytoplasmic expression in a wide variety of cell type from a wide variety of tissues. And that's how it really learned to recognize protein expression pattern. And that's what allows us to be both accurate and robust, because it recognizes the marker expression pattern. It is much more robust to the problems that I mentioned earlier, whether it's background noise, segmentation errors, projection from neighboring cells. It allows us to be a lot more generalizable because of the power of deep learning and because of the normalization that we use, which is per channel per slide. It really enables us to compare between slides and batches across diseases, indications, and imaging modalities. And that's what allows us to be scalable and marker agnostic. So for each project, we use the same deep learning model. We don't need to retrain, and that really enables us to shorten turnaround times. And for a new panel that we didn't see before, it usually, even if it's a high-plex panel on dozens of images, we can return results even less than a month. But if it's already a panel that we trained and already defined all of the parameters that are needed, for example, for the cell typing identification and perform the QC, then we can return results within days, which allows clinical applicability of our pipeline. Now, so as I mentioned, there are many advantages for DL-based protein expression quantification. And here you can see example of 6 data sets and hundreds of markers, both from different imaging modalities, whether it's the cohort a cohort imager, or a Luna-4 comet. There's a wide variety of markers. A lot of them were not seen by the model. Also, during our training of the model, we kept within our both validation and testing set markers that are not seen by the model, and also tumor indications, disease indications that are not seen by the model. And you can see that overall the performance across all of these were very good, with a F1 score above 75% and balance accuracy above 90% in more than 90% of protein. And this is an example of our own multiplex imaging viewer, where you can see all the results. You can see both the markers and apply all the various dynamic ranges. You can see the cell typing result, the neighborhood, the area identification, and even the single marker positivity. And you can see how can we identify cells even within, this is a B cell aggregate, which has several immune cells within it. And you can see that we are able to identify the CD8 T cells, even though there is a lot of overlap with the CD20. This is an example from a Luna-4 comet 28-plex melanoma cohort that we analyzed. And you can see also where we allow the visibility of the single channel protein positivity. And again, you can see which cells within the tertiary lymphoid structures and within the tumor are proliferating and are Ki67 positive. Now we go for, we'll demonstrate an example of our application of our multiplex imaging analysis pipeline for identification of novel biomarker discovery within both non-small cell lung cancer and head and neck squamous cell cancer patients. In both of these indications, only minority of patients benefit from immune checkpoint blockade therapies. And we specially profiled the tumor immune microenvironment and metabolic activity of first-line immune check blockade treated non-small cell lung cancer patients and head and neck squamous cell cancer patients to identify biomarkers associated with treatment response. And that was done in collaborations with Dr. Arutha Kulasine lab in Queensland, in the University of Queensland, and with Dr. David Ream's lab in Yale University. And we used a 45-plex panel, and it was run on a Coya phenocycler that included both immune cell lineage markers, but also a lot of immune cell state markers, and uniquely for this cohort, also a large metabolic markers panel that allows us to perform metabolic phenotyping of the different tumors. Then we applied our MIF analysis pipeline that was the same for both the non-small cell lung cancer and head and neck squamous cell cancer patients, in which we took the MIF image, we performed cell segmentation, then performed cell typing, identifying around 15 different cell types. We then identified spatial neighborhood, and we performed area assignment, including cell state phenotyping. And uniquely for this panel, because we had a lot of both immune and metabolic markers, what we did is that we took our supervised cell typing approach, which allows us to be very accurate in our cell typing. And then within each different cluster, we performed unsupervised subtyping to really identify the different subtypes within each cell type. And then we combined all of these inputs, including the cell type and cell subtype, the phenotypic state, and the area and spatial context, to calculate spatial features, which included the cell distributions and including their subtype and phenotypic state within the different areas, cell and receptor ligand interaction, and protein co-expression features. Now, first, we're going to focus on the non-small cell lung cancer cohort. results, in which we had 72 patients with available relevant clinical outcome data. And in here, we really, because these are tumor microarrays, which were focused mostly on the tumor core, really focused on the tumor metabolic phenotyping and the tumor area in order to perform our biomarker discovery. And as I mentioned before, we took all the identified tumor cells, and then we performed unsupervised clustering based on their relevant phenotypic markers. And as you can see here, we identified 5 different tumor cell subtypes. The first subtype was enriched for CD44. We had a subtype that was enriched for G6PD, a subtype that had a high HEX1 positivity, a subtype that had high HLA positivity, and as you can see, this is a subtype that also had the highest PD-1 positivity, and then another subtype which was rather low in oxidative phosphorylation proteins such as succinate dehydrogenase or citrate synthase. And then we took these 5 metabolic subtypes, and for each tumor, we assigned a subtype based on the majority cell type that it had. And then we compared a few measurements between them. First of all, the different markers, single marker distribution. And we can see that there is distribution within the markers that I mentioned, different marker distribution in the markers that I mentioned before. But there are also other markers which are affected, such as PD-1, which is higher in the HLA positive tumor subtype. We can see that there's also a difference in proliferation, difference in IDO1 positivity, in in CD8 positivity, and in other markers as well. We also saw difference in cell type distribution, where within the HLA positive and CD44 positive subtypes, we had more CD8 T cells, and we saw some more fibroblasts within the different, whether it's the tumor oxphos or the HEX1 tumors as well. We also saw the difference in their clinical outcome based only on the tumor subtype, where the CD44, the tumor CD44 subtype had improved progression-free survival versus the G6PD enriched and HEX1 enriched phenotypes, which had worse clinical outcome. And this is just an illustrative images of the marker distribution within the different tumor clusters, representative tumors within each cluster. And what we can see, which is interesting, is again how important is the spatial context. Because, for example, we can see CD44 positivity in all of the tumors, but only in the tumor CD44 subtype, we can see it positive within the tumor cell. In all the rest of the tumor clusters, it's actually positive within the immune and stromal cells. And that's why spatial context is so important when we phenotype tumors. Next, once we had this subtype, we did our spatial feature calculation. As I mentioned, we combined the phenotypic markers, cell typing, cellular neighborhood, and area assignment, calculate both cell and cell subtype fractions within areas, combined with phenotypic states, cell-cell interactions, including ligand receptor interactions, protein co-expression quantification within different cell type, for example, for identification of terminally exhausted CD8 T cells, which are positive for both PD-1 and LAG3. And then the next thing that we checked is actually we wanted to see within each tumor subtype, which were the main features that were associated with progression-free survival. And the way that we did that is actually we thresholded each spatial feature in an unsupervised manner, so it would not be exposed to the outcome. So it'd be less biased approach for dividing the spatial features into binary groups. And then we looked on what are the top features that are associated with response within each subgroup within the tumor area. And then we looked on their significance within the tumor subtype and compared them also in all the other subtypes. And what we can see that there are definitely some features that are more that are both relevant for the subtype and for the general population, but most of the features are only relevant for the certain subtype. So for example, if we look on LAG3 positive CD8 T cells in the tumor, they are very significant within the tumor CD4 subtypes, but not as significant across the cohort, versus the CD4 positivity within the tumor was also associated with better response across all the cohort. Another thing that we can see from this analysis is that within some subtypes, the features that are associated with the response are immune-related, as expected for immunotherapy outcome. For example, within tumor CD44, we can see the distribution of CD4 positive macrophages, the CD4 positivity, and CD8 T cells, all associated with better overall response. Same with the tumor HLA subgroup, where we see that HLA positive CD8 T cells, tumor cells, and macrophages were the features that were mostly associated with response. But actually within the more classic metabolic subtypes, such as the tumor G6PD subtype or the tumor HEX1, what we can see is that the features that were associated with treatment outcome were actually associated to other metabolic function of the tumor and not for the immune markers. For example, in the tumor G6PD subtype, we can see that the positivity for ATPA5, which is a part of the oxidative phosphorylation chain, was predictive for response. Whether if you add also HEX1 tumor cells, which were 1600 hydrogenase high positive, it was associated with worse clinical outcome. And we can see here the importance of the metabolic phenotype of the cells in order to better predict clinical response within these subgroups. Another analysis that we did is we looked on what are the most differentially expressed features in all the cohorts, and then we looked to see which of the tumors were the ones that affected their positivity. And what we saw is actually that all of the top features were mainly associated to a specific tumor subtype. So again, for this analysis, we did not use the different tumor subtypes. We just took all the samples, looked on what was the most differentially expressed spatial features. And for example, we can see that the CD44 was associated with better overall response. But if we take out all the CD44 positive tumor subtype, we actually see that in all the rest of the samples, it doesn't really affect the responsiveness. Same for PD-1 positivity. We can see that it mostly segregates between the responders and non-responders in the HLA group, much less, and some also in the CD44 group, much less in the other subtypes. So we can see that PD-1 positivity is only relevant for several tumor subtypes and not to all of them. Same for the HEX1 tumor cells HLA, which was mostly relevant for the HEX1 subgroup, or the G6PD tumor cells, that their ASKT2 positivity was associated with a non-clinical benefit. But again, it's only mostly within the G6PD subtype. If you look on the rest of the subtype, this feature is not very relevant. And when we, so in order to emphasize the importance of subtyping, in order to accurately predict the response, we also took the t-statistics of all the spatial features within the subtypes, and we correlated between the t-statistics. And what we show that for most subtypes, the correlation was pretty low between the different t-statistics. Again, emphasizes the importance of subtyping for accurately predicting response for immunotherapy. So to really summarize our findings, the next step that we did was to develop a tumor subtype-based multivariate immune check blockade outcome predictor. And the way we did that, we first split the data into trained and independent test set. And within the training set, we gave the model the subtype of each sample and the top spatial features that was associated with the response within these subtypes, and then we combine these to develop our multivariate predictor. And what we see, first of all, is we have a very nice area under the curve within the test set with AUC of over 0.9 and a very nice p-value, where within the predicted response group, we had around 80% response rates versus in the predicted resistance, we had only a 20% response rate. And these are already numbers which are clinically applicable and are a lot higher than currently used PD-L1 biomarker for patient certification. Next, we aim to profile our head and neck squamous cell cohort. You can see also another benefit of multiplex imaging is it's a kind of a Rorschach test. And in this case, we performed the same pipeline. We had 65 samples with relevant clinical data. And we used the same methodology to identify tumor subtypes. And actually, interestingly, we found quite similar subtypes. There was some difference in the CD44 positivity, which also, in this case, included the HLA-A high group. So they were both HLA-A high and CD44 positive. But other than that, we also found the G6PD tumor cells enriched group, the HEX1 enriched group, and the OXPHOS negative group as well. So here we had 4 tumor clusters. And again, we compared the difference in the different distribution of single channel markers. And as you can see here, we also identified immune-related markers that were higher within the CD44 positive group, whether it's PD-L1, LAG3, IDO1, for example. And interestingly, for example, FOXP3 was enriching the OXPHOS negative. We also saw difference in the distribution of cell types within the different groups, which again, with upregulation of CD4 T cells, CD8 T cells, and to some extent macrophages within the CD44 group, but also these cells were elevated. So we had higher macrophages content and higher Tregs content within the OXPHOS negative group. And we saw also difference in the clinical benefit rate, which was high in the tumor CD44 in HEX1 group. And we can see also they had difference in the progression-free survival, which was higher in these groups and lower in the G6PD and OXPHOS negative groups. I want to add that relatively the numbers, as you can see here, were lower. We had lower presentation of the G6PD and OXPHOS group. These were in total 13 patients, so we cannot do the prediction of the biomarkers within this spatial subgroup. So we focused on the CD44 and HEX1 group, which were larger, to really identify which spatial features were associated with response within these groups. And again, what we showed is that they actually had specific spatial features that were associated within response within each tumor subtypes. But when you apply them to all other subtypes, you can see that they are not really significant. So it again shows us the importance of the metabolic phenotyping in identification of the different tumors subtypes in order to have a much more accurate biomarker identification. And with that, I want to go to our last polling question, which is, what do you see as the main barriers to adopting multiplex immunofluorescence in biomarker discovery? This is a multiple choice question, and the options are high cost of experiment, limited throughput and scalability, slow turnaround, few samples per run, complexity of image and data analysis, technical variability in staining and imaging, and lack of standardized workflows or other. So please vote. So I see the majority of you mentioned mostly two reasons, which is the complexity of images and data analysis and the high cost of experiments. And we can say that here at Nucleai, we're really focusing on simplifying the complexity of image and data analysis. And we're also working with multiple partners to really enable also the technical part of the MIF image capturing, really apply it in large scale for both biomarker discovery and in the future for clinical application. With that, I would like to summarize and conclude. So in this talk, we show there's a growing need for spatial biomarkers, which can help treatment decision-making. We show the potential of MIF to revolutionize biomarker discovery in precision medicine, but we also talked about the limitation and how it's currently hindered by inefficient staining procedures and analysis tools, which we aim to improve. We developed a deep learning-based analysis pipeline that demonstrated robustness across markers, tumor indications, and imaging modalities without the need to retraining, which allows us to really apply it in large scale and even in a clinical setting. And we showed the example and the importance of applying this MIF pipeline to really get deep insights into the response mechanism of patients to immunotherapy. In this case, we showed the importance of tumor cell metabolic subtyping in both non-small cell lung cancer and head and neck squamous cell patients to really define distinct tumor clusters that differed in the clinical outcome, but also had very different spatial features that's associated with response within each subtype. And thus, this is crucial to have a good enough diagnostic test that can really help us navigate patients both that can benefit from immunotherapy. So I hope that in this talk I highlight the importance of this metabolic profiling to outcome prediction and why it's important for biomarker discovery. I would like to thank both of our collaborators who participated in this project and the whole of the Nucleai team, which worked very hard to develop all of these amazing algorithms. And with that, I would like to conclude and open the stage for questions.
Great, thanks, Itai. Thank you for that fantastic and informative presentation. Now everyone will move into our Q&A session. We have already received some great questions, which we'll get to in a second. But if you haven't submitted yours yet, please feel free to drop it in the Q&A box, and we'll get to as many as we can. If we don't get to it, we'll try and do a direct follow-up as well. So Itai, the first question, where do you see the greatest importance of multiplex immunofluorescence in drug discovery and clinical development process?
Yeah, so I think multiplex immunofluorescence can actually benefit all stages of clinical development, whether it's from drug discovery. We have conducted several, for example, prevalence studies to really have a large-scale, accurate quantification of protein expression across diseases. And that can really help you with how to strategize your drug development and which, for example, if you want to have a bispecific target, to quantify co-expression of proteins within different tissues. It can be also applied within early clinical development, phase 1, phase 2, for example, if we want to do protein scoring for patient selection, or to really start accumulating the data for phenotyping of the different patients in order to really have a more robust biomarker approach for our later stages. And we also apply the way we use MIF for more advanced clinical trials is that we take all of the insights that we get from the MIF platform, and we know that currently it's quite hard to apply it in a large-scale study. So in Nucleai, we developed a complexity reduction methodology, which aims to really simplify the panel and to really use both H&E, IHC, multiplex immunohistochemistry, or low-plex panels. So to really simplify the prediction model, so it will need the minimum number of elements, but will still keep its prediction ability. So it can be applied all across the studies, and of course also for retrospective studies where we want to have the biological insights that will allow us to develop a clinical diagnostic assay.
Great, thanks, Itai. Next question, how do you see this platform applying to novel targets?
It's a great question. So the way we developed our pipeline is that it's marker agnostic. So if we think about it, actually, let's say we talk about targets either expressed by immune cells or by tumor cells, most of the expression patterns are pretty similar, meaning they either will be membranous, cytoplasmic, nuclear, or some combination of the above. And we trained our model on a very wide variety of markers, so it really learned to be marker agnostic. So and we validated our model on unseen markers. In all of them, we did not see a drop-off of the model performance. So we can use our MIF analysis pipeline on novel targets without any issue, even within novel disease types. Because we define supervised methods to identify cells, so from project to project, what we will adjust is the cell rules, how to identify the cells. We can adjust our area methodology. For example, let's say we're now within inflammatory bowel disease. Instead of tumor areas, we can identify, we have different definitions to identify the crypts, the lamina propria, or the submucosa. But the core of our technology, which is the protein expression, will be the same. And it's transferable and marker agnostic, so it won't be an issue.
Great, thanks, Itai. Next question, how many markers do we typically use for this cohort and why?
So it really depends on the clinical question and the phase or the scale that you want for your study. So I can give a few examples. Let's say we want to develop now a multiplex imaging screener for different, let's say, ADC targets and PD-1 expression to really quantify our current approved clinical diagnostics in a way that will help us screen patients and navigate them to the appropriate diagnostic. So this is a screener concept in which we take the available or current targets or approved drugs, we quantify them, and then we aim the patient, which later goes and have the formal diagnostic. And the goal for that is really to help with navigating the patient and to preserve tissue, because we know that the biopsies are getting smaller and smaller. So tissue is the issue because we need them for DNA testing and for other biomarkers. So we try to preserve as many tissues as possible. So for this example, we need quite a low number of markers. If it's a discovery cohort, we usually will try to go deeper, really have the deep, meaningful insights that we can have from several kinds of cell types and immune cell types. So we can definitely go to the 40, 50, even 60 plex. For us, for analysis, we can handle all of it, even 100 plex, if someone provides us that. But then when we go to the more, let's say, large cohorts, advanced clinical setting, usually we'll focus on a low plex and a low number of targets, best designed after we did the deep profiling and then had a really kind of informed decision making in choosing the markers for the low-plex for the advanced clinical setting.
Thanks, Ty. We got time for one or two more questions. Next question is, do you believe that MIF-based biomarkers will be adopted in routine clinical practice in the future?
So I do believe yes. This is my strong belief, namely because, and mostly in the screening application, just because clinicians will need help with navigating the patient, even once we will have, let's say in the multi-treatment era, where there will be several treatment options in order to preserve tissue and really to navigate the patient to the correct treatment. It will be best to use MIF to capture multiple clinical assays on the same panel, or clinical targets on the same panel, and then navigate the patient to the right clinical diagnostic assay. And I believe that will be inevitable in the future, just because we'll have multiple treatment options, and we need a tool that will help us make clinical decision making. And because spatial biology is so important to the new era of treatment, I believe that MIF will have a role in that. We are almost ready from the algorithmic perspective to do that. We're still waiting for the right partner or development of assays that would allow us to do it in a high throughput manner, the staining part.
Great. And last question, do you apply AI algorithms for biomarker discovery in modalities outside of MIF?
Yes, we definitely do that as well, both for H&E and for IHC. We have our own deep learning based protein scoring for antibody-drug conjugates, where we can accurately quantify the expression within the different cell compartments. And we show, and we actually had a clinical trial with a pharma company that we'll hope will be published later this year, where we show that AI-based scoring was able to separate the patients quite nicely, whether pathologist-based scoring was not able to separate between the responders and non-responders to the novel drug therapy.
Great. With that, I want to thank everyone for joining us today. That will conclude our session. If we didn't get to your question, I do apologize, but don't worry, we'll follow up directly after the webinar. A link to the recording will also be shared via email, so please keep a lookout for that in your inbox. And again, thank you, and you have both Atai and my contact, and hope to talk with you soon. Have a great day.